Hello all, welcome to the first session on food microbiology. In this lecture, I'll be throwing some light on an important topic called food poisoning and its importance and relevance for the food industry. Microbiology is an interesting science, and microbiology with respect to food is even more fun due to its positive and negative effects. So let's get started. In this lecture, we will be covering the following topics: definition of microbiology and food microbiology, types of food poisoning and its definition, what mechanisms are in place to control food poisoning, main pathogens associated with food infection, presence in food products and their control. Food is considered to be one of the basic needs of life. In present century, since both parents are working, there's an increasing demand for convenience food, meaning a food which is ready to eat. This has led to emergence of a new discipline of microbiology called as food microbiology. Microbiology as such deals with organisms that are not visible to the naked eye, but are omnipresent. Therefore, food microbiology deals with not only understanding microbes causing spoilage and poisoning, but also methods to control and prevent spoilage and poisoning, so that we can extend the shelf life of foods. It forms basis for subjects like thermal preservation, food processing, and food formulation. Coming to microbes associated with food, it can be divided into three categories. Pathogens are the ones causing poisoning. Then you have spoilage organisms, and finally you have beneficial organisms. What are foodborne pathogens? Foodborne pathogens are those microbes which are present in food and they are responsible for causing a disease or symptom of a disease in the host. Now, why is it essential to study food poisoning? It's necessary to understand the concept of food poisoning, as every year around 600 million people are affected, causing around 400,000 fatalities. Now, already food industry is taking the necessary precaution, but at times food poisoning is result of mismanagement on the end of consumers. Types of food poisoning. Depending on what is causing the disease or symptom of a disease in the host, food poisoning can be categorized into three. Food infection, which is caused by uptake of a low number of microbes. Food intoxication, which is caused by uptake of food containing microbial toxins. And finally, you have food toxi-infection, which is caused by uptake of high number of microbes. And this high number of microbes produces toxin in the host, which is responsible for causing disease or its symptom. So far, we have spoken about food poisoning. Now, one must be thinking what effective mechanisms are in place to control both food infection and intoxication. So here, if you look at the middle round or the circle, it represents controls that are common for both types of food poisoning, that is food infection and food intoxication. So what at industrial level, what is done is that first there is quality check of raw material, because many microbes are omnipresent in nature. Understanding what is the microbial load of raw material will give you an insight on what kind of processing step needs to be done for that particular food product. Secondly, post-contamination to be prevented, and in case it occurs, there should be measures to control it. Thirdly, processing conditions need to be respected. So when we are preparing food, convenience food, we are going to subject it to heat treatment, cooling, and then probably modification of several other intrinsic and extrinsic factors, so that we get a product with extended shelf life. So whatever is the step that we are taking, we need to respect it. We should follow the time-temperature combination. Focus on storage and supply chain, because we are reducing food with extended shelf life. Then finally, following good agricultural practices, good hygienic practices, good manufacturing practices, and HACCP principles. HACCP is hazard analysis and critical control points. I will cover this topic later in one of the other lectures. Apart from that, you have to take several precautions for both types of poisoning. So when you're doing quality check for raw materials, in case of food infection, you can do indirect testing using hygiene indicators, which will indicate the presence of pathogen. This is cheaper testing methodology and it's easier because you get results within few hours. In case of direct testing for pathogen, it's a long, tedious process. But if you are very well aware of what pathogen is present in what type of food, then you can go for this kind of direct testing. And if you have time in the world, then you have to give importance to time-temperature combination at all stages, be it processing, be it storage, or be it during supply chain. And finally, cool condition, that is cold chain, needs to be respected. In case of food intoxication, you see that during quality check of raw materials, it's not possible to completely reduce the microbial load, but you can set a target value and see whether it is within the tolerable limit. Then, the food that is being produced, mostly canned food products, they are subjected to fast cooling, so the time temperature should be maintained here. Then packaging, they can be subjected to modified atmospheric packaging or controlled atmospheric packaging. And finally, storage. During storage, if there are any changes in acidity of the food product, or for that matter, the atmosphere in which the food is placed, or water activity, then it needs to be reported and monitored. Finally, some of the canned food products like sea fish and seafood, especially, they undergo processing like smoking, fermentation, drying. That time it is of immense importance that you take into account the proper time-temperature combination that is required to reduce the microbial load. Let's talk about the first type of food poisoning, which is called as food infection. Food infection is caused by uptake of a low number of microbes. Now, this microbe, they disrupt the normal flora of the intestine, meaning they are readily able to invade the host body. What type of hosts are susceptible to food infection? Young, old, pregnant, and immunodeficient. Here, in case of food infection, the microbe itself is responsible for causing disease or its symptoms in the host. The big four associated with food infection, especially relevant to Europe and the US, you have Listeria Monocytogenes, Salmonella, Campylobacter jejuni, and E. coli. Listeria monocytogenes is a gram-positive bacteria that causes listeriosis. Common symptoms include fever and muscle pain. Sometimes you do have nausea and diarrhea. People belonging to young, old, pregnant, and immunodeficient group are highly susceptible to this pathogen. So what are the characteristics of this bacterial cell? They are said to be psychotropic in nature, meaning they can resist cold temperatures. Some studies show that this organism is capable of growing at temperatures of 0 degrees Celsius. They are also resistant to unfavorable preservation conditions, meaning acidic pH or low water activity containing products. Now, what causes Listeria outbreak? First, post-heating contamination. When I say post-heating contamination, I mean if there is a product that has been processed, then the product is open for cutting or repackaging, then it is susceptible to Listeria outgrowth. Secondly, cleaning and disinfection. If it is not followed as per industry standard or norm, and one is not following good work practices, then the product is susceptible to Listeria outgrowth. Food products that are related to Listeria outbreak include preserved food products with extended shelf life in the refrigerator. Example, you have raw milk, soft cheeses, cold smoked salmon, sliced packaged meats, then fresh fruits, vegetables, meat, fish. Now, having said that it's the most heat-resistant vegetative pathogen, we have to ensure that it's absent per 25 gram of food product. In order to ensure that it is absent per 25 gram of food product, we subject it to pasteurization temperature of 70 degrees Celsius for 2 minutes, which brings in about 6 log reduction in the count of bacterial cells. Controlling and respecting cold chain is another way to ensure that there is no outbreak of Listeria. Next, we have Salmonella, which is a gram-negative bacteria which causes salmonellosis. Salmonellosis is characterized by abdominal cramps, fever, diarrhea. Now, this organism is active between 8 degrees Celsius and 45 degrees Celsius, meaning it cannot grow at refrigeration temperatures. Then it can grow in low water activity containing products. So now what is the cause of Salmonella food poisoning? It is due to consumption of raw or insufficiently heated eggs or food that contain raw eggs. The other source is pork and poultry meat. Food products containing eggs, like for instance, you have bakery products, you have your croissant, cake, then you have fresh meat and poultry. Milk sometimes, because the same facility has both dairy products as well as poultry. So as a result, it is quite possible that milk is contaminated with salmonella because of some cross contamination. And then main source is your egg. So what mechanism is in place to control this? What is aimed for at industry level is absence of this pathogen for 25 gram. So this is done by following good manufacturing practice, good hygienic practice, good agricultural practice, and then you will follow HACCP, which is hazard analysis critical control point. The product is heated to 70 degrees Celsius for about 2 minutes, which is the pasteurization condition. And then finally, ensuring that we follow the cold chain rules. The third organism, Campylobacter jejuni, it is responsible for causing a condition called as campylobacteriosis, which is featured by fever, abdominal cramps, and diarrhea. These organisms, they are said to be thermotolerant. They are very active between 30 to 45 degrees Celsius, but cannot grow at beyond 50 degrees Celsius and lower than 7 degrees Celsius. Now, what is responsible for causing Campylobacter food infection? It is caused by consumption of undercooked poultry, cross-contamination when you have dairy and poultry in the same farm. It's potential that milk is also contaminated with the organism, and then insufficient heating of poultry dishes. Food products that are susceptible to this microbe, you have poultry meat, mainly poultry meat. 30 to 40% of the time, it is the poultry that carries this organism. Then raw milk, and sometimes your seafood crustaceans. Control at an industrial level is this. One, you have to keep in mind, one cannot aim for Campylobacter-free poultry. It's not at all possible. So there is a minimum limit, which is 100 colony forming unit per gram for poultry. This is achieved by following good manufacturing practices, good hygienic practices, good agricultural practices, and HACCP principles. Also, what temperature is used for controlling this organism is the same pasteurization temperature, that is 70 degrees Celsius for 2 minutes, because the most heat-resistant vegetative cell is Listeria Monocytogenes. So other organisms are less resistant to heat compared to Listeria vegetative cell. You may find it surprising to find this organism in the list of pathogens. It is true that this microbe is found in the intestine of human beings and animals, but there are several strains that are considered to be pathogenic as they carry virulence factors. Based on the virulence factor, E. coli can be divided into 5 types. You have entero-aggregative E. coli, entero-hemorrhagic E. coli, entero-invasive E. coli, enteropathogenic E. coli, and enterotoxigenic E. coli. Some of the E. coli strains, they can produce toxins. That's what I've written under characteristics of E. coli. And this organism is active between 4 and 45 degrees Celsius, meaning it can affect both products that are stored at normal room temperature and that which are stored in cold conditions. Now, what causes E. coli outbreak? If you ingest undercooked food, so any bacterial infection or any food infection or food poisoning case, it can be result of ingestion of undercooked food. Secondly, ingestion of improperly washed fruits and vegetables. Now, you may be wondering, why is this the case? Because E. coli is used as fecal indicator. So this is an organism which is omnipresent. Therefore, one has to ensure that the amount of pathogen present is controlled right from farm to fork. So, because E. coli is fecal hygiene indicator, it can be used as hygiene indicator during production, storage, processing, but it cannot grow in cold conditions. Very cold conditions, it cannot grow. So if you want to carry out some sort of check to ensure that this organism is not there or any other organism or pathogen is not there, then you can check for E. coli in the product. And it will show to what extent GAP, that is good agricultural practice, has been followed. Because of the low infective dose and high severity of the disease, what we aim for is absence per 25 gram. This can be achieved by following good manufacturing practices, good hygienic practices, good agricultural practices. Then 70 degrees Celsius for 2 minutes. This is the pasteurization condition. Cell cannot withstand this. An indicator during production, processing, and storage. Some of the food products that are associated include leafy vegetables because they are grown at a low level in the ground, raw milk, sprouted seeds, and then you have minced meat, fresh meat, or meat products. In the next lecture, we will be discussing about food intoxication and food toxi-infection, pathogens associated with food intoxication and toxi-infection. Merci beaucoup.